हेलो फ्यूचर डॉक्टर्स, माय नेम इज़ कीर्ति और मैं आपको हर क्लास में एनसीईआरटी लाइन बाय लाइन डिकोड करती हूँ। मैंने एक आइडिया लगाया, जिससे आप कोचिंग से घर जाते हो और घर से कोचिंग जाते हो, इस बीच आपका बहुत टाइम वेस्ट होता होगा ना। इसलिए मैं आज से एनसीईआरटी ऑडियो बुक रिकॉर्ड कर रही हूँ। आते जाते टाइम आप इसको सुन लेना। तो आज की क्लास में हम पढ़ने वाले हैं सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट। तो आइए बिना टाइम वेस्ट किए स्टार्ट करते हैं। Are we not lucky that plant reproduce sexually? The myriads of flower that we enjoy grazing at, the scent and the perfumes that we soon over, the rich colors and attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structure and the inflorescence flower and the floral parts shows an amazing range of adaptation to ensure formation of the end product of sexual reproduction, the fruits and the seeds. In this chapter, let us understand the morphology, structure and the process of sexual reproduction in flowering plant. Angiosperm 2.1. Flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flower since time immemorial. Flowers are object of aesthetic, ornamental, social, religious and cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, sadness, grief, mourning, etc. At least 5 flowers of ornamental value that are commonly cultivated as home and in gardens. Find out the name of 5 flowers more than used in a social and cultural celebration in your family. Have you heard of floriculture? What does it to refer? To a biologist, flowers are morphological and embryological marvels and sites of sexual reproduction. In earlier class, you have read the various part of flower will help you recall the parts of typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? 1.2. Pre-fertilization, structure and event. Such before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structure, the endoecium and the gynoecium, differentiate and develop. You would recollect that the endoecium consists of a whole of stamen representing the male gamete reproductive organ and gynoecium represent the female reproductive organ. 1.2.1. Stamen, microsporangia and pollen grain. Show the 2 parts of typical stamen, the long and cylindrical stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus of the petal of the flower. The number and the length of stamen are variable in flower of a different species. If you were collected to stamen each 10 flower, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size and in nature. Carefully observation of each stamen under a disgusting microscope and making a neat diagram, you would the range in shape and attachment of anther in a different flower. A typical angiosperm anther is a bilobed with each lobe having a 2 theca. They are diathecious, often a longitudinal grooves run lengthwise separating the theca. Let us understand the various type of tissues and their organization and in transverse section of the anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a 4-sided tetragonal structure consisting of 4 microsporangia located at the corner, 2 in each lobe. The microsporangia develop further and become pollen sac. They extend longitudinally all through the length of the anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near a circular in outline. It is generally surrounded by a 4-wall layer the epidermis, endothelium, middle layer, and the tapetum. The three outer wall layer perform the function of protection and help in dehiscence of the anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Shells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think how tapetal cells could become binucleated? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. As the anther develop, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. What would be the ploidy of the cells to be tetrad? As each cell of the sporogenous tissue is capable to giving rise to microspore tetrad. Each one is potential pollen or microspore mother cell. The process of formation of microspore from pollen mother cell PMC through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of 4 cells. The microspore tetrad, as the anther mature and dehydrate. The microspore dissociated from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represent the male gametophyte. If you touch the open anther of hibiscus or any other flower, you would find the deposition of yellowish powdery pollen grains on your finger. Sprinkle these grains on a drop of water taken on a glass slide and observe under microscope. You will be really amazed at the variety of architecture, size, shape, color, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It is a prominent two-layered wall. The hard outer called exine is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperature and strong acid and alkyl. No enzyme that grades sporopollen is so foreknown. Pollen grain exine has prominent apparatus called germ pores where sporopollen is absent. Pollen grains are well preserved as fossils because of the presence of sporopollen. The exine exhibit a fascinating array of patterns and designs. What do you think? The exine should be hard? What is the function of germ pore? The inner wall of pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative and the generative cells. The vegetative cell is bigger, has abundant food reserve and a large irregular shaped nucleus. The generative cell is small and float in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grains are shed at this 2-celled stage. In the remaining species, the generative cell divide mitotically to give rise to the 2 male gametes before pollen grains are shed 3-celled stage. Pollen grains of many species can serve allergic and bronchial affliction in some people, often lead to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grasses that come into India as a contaminant with import wheat has become in occurrence and cause pollen allergy. Pollen grains are rich in nutrition. It has become a fashion in recent years to use pollen tablets as food supplement in Western countries. A large number of pollen products in the form of tablet and syrup are available in market. Pollen consumption have been claimed to increase the performance of athletes and race horse. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bringing about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grain remain viable is highly variable and to some extent depend on the prevailing temperature and humidity. In some krills, such as rice and wheat and pollen grains, lose viability within 30 minutes of their release. And in some members of Roaceae, Leguminosae and Solanaceae, they are maintained viability for months. You have here or for storing stem and sperm of many animals, including human, for artificial insemination. It is possible to store pollen grains of a large number of species for a year in liquid nitrogen minus 196 degrees Celsius. Such stored pollen can be used as a pollen bank, similar to seed bank in a crop breeding program. The pistil megasporangium, ovule and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil monocarpally or may have more than one pistil multicarpally. When there are more than one, the pistil may be fused together syncarpous or may be free apocarpous. Each pistil has a three part: stigma, style and ovary. The stigma serves as a landing platform for pollen grains. The style is an elongate cylinder part beneath the stigma. The basal bulge part of the pistil is the ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11th. Rising from the placenta are megasporangia, commonly called ovule. The number of ovule in an ovary may be 1, wheat, paddy, mango, or too many, papaya, watermelon, and orchid. The megasporangium ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fused with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has a one or two protective envelope called integument. The integument encircles the nucleus expected at the tip, where a small opening called the micropyle is organized. Opposite the micropylar end is the chalazal, representing the basal part of the ovule. Enclosed within the integument is a mass of a cell called the nucleus. Cells of the nucleus have abundant reserve food material located in the nucleus in the embryo sac or female gametophyte. An ovule generally have a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovule generally differentiate a single megaspore mother cell MMC in the micropylar region of the nucleus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspore. Female gametophyte. In a majority of flowering plant, one of the megaspore is a functional, while the other 3 degenerate. Only the functional megaspore develop into the female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cells of the nucleus, MMC, the functional megaspore and female gametophyte? Let us study about the formation of embryo sac in detail. The nucleus of the functional megaspore divide mitotically to form 2 nuclei which move to the opposite pole, forming the 2 nucleated embryo sac. 2 more sequential mitotic nuclear division result in the formation of 4 nucleated and later the 8 nucleated stage of the embryo sac. It is interesting to note that this mitotic division are strictly free nuclear, that is, nuclear division are not followed immediately by cell wall formation. After the 8 nucleate stage, cell wall are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac. 6 of 8 nuclei are surrounded by cell wall and organized into cells. The remaining 2 nuclei, cell polar nuclei, are situated below the egg apparatus in the large central nucleus. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have a special cellular thickening at the micropylate tip called filiform apparatus. Called filiform apparatus, which play an important role in guiding the pollen tube into the synergy. 3 cells at the chalazal end and are called antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at the maturity through 8 nucleated is 7-celled. 1.2.3 Pollination. In the preceding section, you have learned that the male and female gametes in a flowering plant and produce in the pollen grains and embryo sac respectively. As both of type of gametes are non-motile, they have to be both together for fertilization to occur. How is this achieved? Pollination is the machines to achieve this objective. Transfer of pollen grain sheet from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agent to achieve pollination. Can you list the possible external agent? Kind of pollination. Depending on the source of pollen, pollination can be divided into 3 parts. First is autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. In a normal flower which open and expose the anther and the stigma, complete autogamy is rather rare. Autogamy in such flower require synchrony in pollen release and stigma respectively, and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as a viola, common pansy, oxalis and comelion produce 2 types of flower, chesmogamous flower which are similar to flower of other species with exposed anther and stigma, and chilistogamous flower which do not open at all. In such flower, the anther and the stigma lies close to each other when anther dehisces in the flower bud. Pollen grains comes in contact with the stigma to effect pollination. Thus, chilistogamous flowers are invariable autogamous as there is no chance of cross-pollination leading on the stigma. Chilistogamous flower produce assured seed set even in the absence of pollination. Do you think that chilistogamous is advantage or disadvantage to the plant? Xenogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although geitonogamy is the functional cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to the stigma of a different flower. This is the only type of pollination which during pollination brings genetically different type of pollen grain to the stigma. Agent of pollination. Plant use 2 abiotic wind and water and 1 biotic animals agent to achieve pollination. Majority of plant use biotic agent for pollination. Only a small portion of plant use abiotic agent. Pollen grains come in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollens when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transferred in wind current. They often possess well-exposed stamens so that the pollen are easily dispersed into wind current, and large, often feathery stigma to easily trap airborne pollen grain. Wind pollinate flower often have a single ovule in each ovary and numerous flower packed into an inflorescence. A familiar example in the cone crab. The tassels you see the nothing but the stigma and the style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is a quite rare in flowering plant and is limited to about 30 genera. mostly monocotyledons, is against this. You would recall what water is a regular mode of transport for the male gametes among the lower plant group such as the algae, bryophyte and pteridophyte. It is believed, particularly for some bryophyte and pteridophyte, that their distribution is limited because of the need for water for the transport of male gamete and fertilization. Some example of water pollinated plant are Valsinaria and Hydrelia, which grow in fresh water, and several marine sea grasses such as Juicera. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as the water hyacinth and water lily, the flower merge above the level of water and are pollinated by insect or wind are in a most of the land plant. In Valsinaria, the female flower reach the surface of water by the long stalk called the male flower or pollen grains are released on the surface of water. They are carried passively by water current. Some of the eventually reach the female flower and the stigma. In another group of water pollinated plants such as the sea grasses, female flower remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water-pollinated species, pollen grains are protected from wetting by mucilaginous covering. Both wind and water pollinating flower are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plant use a range of animal as pollinating agent. Bees, butterflies, flies, beetles, wasps, and moth, birds, sunbirds, and hummingbirds and bats are the common pollinating agent. Among the animal insect, particularly bees and the dominating biotic pollinating agent. Even larger animals such as some primates, lemur, tree-dwelling rodents, or even reptiles, jacogo lizard and garden lizard, have also been reported as a pollinator in some species. Often flower of animal pollinated plant are specially adapted for a particular species of animal. Majority of insect pollinated flower are large, colorful, fragrant and rich in nectar. When the flower are small, a number of flower are clustered into an inflorescence to make them conspicuous. Animals are attracted to flower by color and or no fragrance. The flower pollinated by flies and beetles secrete flue odor to attract this animal. To sustain animal visit, the flower have to provide reward to the animals. Nectar and the pollen grains are the usual floral reward. For harvesting the rewards from the flower, the animal visitor comes in contact with the anther and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal pollinated flower. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral reward are in providing safe place to lay eggs. An example is that the tallest flower of Amorphophallus, the flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant Yekaka, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary and the flower. It turns get pollinated by the moth. The larva of the moth comes out of the egg as the seed start developing. Why do not you observe some flower of the following plant or any other available to you? Cucumber, mango, people, coriander, papaya, onion, labia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You will have to patiently observe the flower over a few days and at different times of the day. You could also try to see whether there are in any coloration in the characteristic of a flower to the animal that visit it. Carefully observe if any of the visitor comes in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as a pollen and nectar robber. You may or may not be able to identify the pollinator, but you will surely enjoy your efforts. Outbreeding device. Majority of the flowering plant produce hermaphrodites. Flower and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination result in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma respectively are not synchronized. Either the pollen is released before the stigma become respective, or stigma becomes respective much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding in self-incompatibility. This is the genetic machines and prevent to self-pollen from the same flower or other flower of the same plant from fertilizing the ovules by inhibiting pollen's germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flower. If both a male and female flower are presented on the same plant, such as a castor and maize monoecious, it prevents autogamy but not zytogamy. In several species, such as a papaya, male and female flower are present on different plants. That is, each plant is either male or female dioici. This condition prevents both autogamy and zytogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma, often pollen or the wrong type either from other species or from the same plant if it is self-incompatible. Also land on the stigma. The pistil has the ability to recognize the pollen, whether it is the right type of compatible or the wrong type of incompatible. It is the right type. The pistil accept the pollen and promote the post-pollination event that lead to fertilization. If the pollen is wrong type, the pistil reject the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection in the result of the continuous dialogues between pollen grains and the pistil. This dialogue is mediated by chemical component of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollens and pistil component and the interaction leading to the recognition followed by the acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grains germinates on the stigma to produce a pollen tube. Through one of the germ pore that contain of the pollen grains move into the pollen tube. Pollen tube grow through the tissues of the stigma and the style and reach the ovary. You would recall that in some plants, pollen grains are shed at two-celled condition, a vegetative cell and generative cell. In such plant, the generative cell divides and forms two male gametes during the growth of pollen tube in the stigma. In plant which shed pollen in three-celled condition, pollen tube carry the two male gamete from the beginning. Pollen tube, after reaching the ovary, enter the ovule through the micropyle and then enter one of the synergies through the filiform apparatus. Many recent studies you have shown that filiform apparatus present at the micropylar part of the synergic guide the entry of the pollen tube. All these events from the pollen deposition one on of the stigma until pollen tubes enter the ovule are together referred to as the pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by the promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction, even in incompatible pollination, to get desired hybrid. You can easily study pollen germination by dusting some pollen from flowers such as a pea, chickpea, and vinaka on a glass side containing a drop of sugar solution about 10%. After about 50 to 30 minutes, observe the slide under the low power lens of microscope. You are likely to see pollen tube coming out of the pollen grains. A breeder is interested in causing different species and often generation combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from the contamination from unwanted pollens. This is achieved by emasculation and bagging technique. If the female parts bears bisexual flower, removal of anther from the flower, but before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flower have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This is called bagging. When the stigma baked flower attain respectivity, mature pollen grains collected from anther of the male parent are dusted on the stigma and the flower are rebaked, and thus the flower allowed to develop. If the female parent produce unisexual flower, there is no need to emasculation. The female flower bud are based before the flower open. When the stigma become respective, pollination is carried out using the desired pollen and the flower rebaked. 1.3 Double fertilization. After entering one of the synergy, the pollen tube releases the two male gametes into the cytoplasm of the synergy. One of the male gametes move towards the egg cell and fuse with its nucleus, thus completely the syngamy. This results in the formation of diploid cell. The zygote, the other male gametes, move towards the two pollen nuclei located in the central cell called fuse with them to produce a triploid primary endosperm nucleus PEN. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two type of fusion, syngamy and triple fusion, take place in an embryo sac, the phenomena is termed double fertilization, an event unique to flowering plant. The central cell after triple fusion become the primary endosperm cell PEC and develop into endosperm, while the zygote develop into an embryo. 1.4 Post fertilization structure and event. Following double fertilization, event of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed post-fertilization event. Endosperm developed precedes the embryo development, while the primary endosperm cell divide repeatedly and forms a triple endosperm tissue. The cells of the tissues are filled with reserve food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is the nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans. Before seed maturation, or it may persist in the mature seed, castor and coconut, and be used during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruits of coconut, and looks for the endosperm in each case. Find out whether the endosperm in persistence in krill, wheat, rice, and maize. Embryo. Embryo develop at the micropylar end of the embryo sac, where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo, though the seed differ greatly. The earliest stage of embryo development, embryogeny, are similar in both monocotyledon and dicotyledon. Despite the stage of embryogeny, in a dicotyledon embryo, the zygote gives rise to the proembryo and subsequent to the globular, heart-shaped and matured embryo. A typical dicotyledon embryo consists of an embryonal axis and two cotyledon. The portion of the embryonal axis above the level of cotyledon is the epicotyle, which terminated with the plumule or stem tip. The cylindrical portion below the level of cotyledon is a hypocotyle that terminate at its lower end in the radicle or root tip. The root tip is covered with a root cape. Embryo of monocotyledon possesses only one cotyledon. In the grasses family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. As its lower end, the embryonal axis has the radicle and root cape enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of the scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordium enclosed in a hollow foliar structure. The coleoptile shock a few seeds in a bottle, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of embryo and the seed. Seed. In angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruit. A seed typically consists of a seed coat, cotyledon, and embryo axis. The cotyledon of the embryo are simple structure, generally thick and swollen due to the storage of food reserve, as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it completely consumed during embryo development. Example, pea and groundnut. Albuminous seed retain a part of endosperm as it not completely used up during embryo development. Example, wheat, maize, barley, castor. Occasionally, in some seeds such as the black pepper and beet, remnant of nucellus are also persistent. The residual persistence nucellus is the perisperm. Integument of ovule hardens through protective seed coat. The micropyle remains as a small pore in the seed coat. The facilitates entry of oxygen and water into seed during germination. As the seed mature, its water content is reduced. The seed become relatively dry, 10 to 15% moisture by mass. The generally metabolic activity of the embryo shown down. The embryo may enter a state inactivity called dormancy, or if favorable conditions are available, moisture, oxygen, and suitable temperature, they germinate. As ovule mature into seed, the ovary develop into a fruit. The transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develop into the wall of the fruit, called pericarp. The fruit may be fleshy, as in guava, orange, mango, etc., or may be dry, as in a groundnut and mustard, etc. Many fruits have a evolved mechanism or dispersal of seed. Recall the classification of fruit and their dispersal mechanism that you have studied in the earlier class. If there any relationship between number of ovules in an ovary and the number of seeds present in a fruit. In most plants, by the time the fruit develop from the ovary, other floral part degenerate and fall off. However, in a few species such as the apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruit are called false fruit. Most fruit, however, develop only from the ovary are called true fruit. Although in most of the species fruit are the result in the fertilization, there are a few species in which fruit develop without fertilization. Such fruit are called parthenocarpy fruit. Banana is one such example. Parthenocarpy can be induced through the application of the growth hormone, and such fruit are seedless. Seeds offer several advantage to angiosperm. Firstly, since reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seed have better adaptive strategy for dispersal to new habits and help the species to consist in other areas as they have a sufficient food reserve. Young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being product of sexual reproduction, they generate new genetic combination leading to variation. Seed is the basic of our agriculture. Dehydration and dormancy of mature seed are crucial for storage of seed, which can be used as food throughout the year and also to raise crop in the next season. Can you have imagine agriculture in the absence of seed or in the presence of seed, which germinate strategy away soon after formation and cannot be stored? How long do the seed remain alive after they are dispersed? This period again vary greatly. In a few species, the seed lose viability within a month. Seeds of a large number of species alive for several years. Some seeds can remain alive the hundreds of years. There are several records of very old yet viable seeds. The oldest is that of lupinus from Arctica tender. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent year of 2,000-year-old viable seed is the dead palm phoenix discovered during the ecological excavation at King Herod Place near the Dead Sea. After completing a brief account of sexual reproduction of flowering plant, it could be worth attempting to compare the enormous reproductive capacity of some flowering plant by asking the following question: how many eggs are present in the embryo sac? How many embryo sacs are present in the ovule? How many ovules are present in the ovary? How many ovaries are present in the typical flower? How many flowers are present on a tree? And so on. Can you think of some plant in which fruit contain very large number of seed? Orchid fruit are one such category, and each fruit contains thousands of tiny seeds. Similar in case in fruit of some parasitic species such as you have seen a tiny seeds of ficus. How large is the tree of ficus developed from the tiny seeds? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the year? 1.5. Apomaxy and polyembryony. Although seeds in general are the product of fertilization of few flowering plants such as some species as asteraceae and grasses have evolved a special mechanism to produce seed without fertilization called apomaxy. What is fruit production without fertilization called? Thus, the apomaxy is the form of asexual reproduction that mimics sexual reproduction. There are several ways to development of apomatic seed. In some species, the diploid egg cell is formed without reduction, division, and develop into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such species, each ovule contain many embryos. Occurrence of more than one embryo in a seed is referred to as a polyembryony. Take out seed of orange and squeeze them. Observe the many embryo of the different size and shape from each seed. Count the number of embryo each seed. What would be the genetic nature of apomatic embryo can then be called clones. Hybrid variety of cereals of our food and vegetable crops are being extensively cultivated. Cultivation of hybrid has increased productivity. One of the problem of hybrid is that hybrid seeds have to be produced every year. If the seeds collected from the hybrid are sown, plant in the progeny will segregate, do not maintain hybrid characters. Production of hybrid seed is costly, and hence the cost of hybrid seed become too expensive for the farmers if these hybrid are made into apomatic. There is no segregation of character in the hybrid progeny, then the farmer can keep on using hybrid seed to raise new crop year after year, and he does not have to buy hybrid seed every year. Because of the importance of apomaxis in hybrid seed industries, active research is going on in many laboratories around the world to understand the genetics of apomaxis and to transfer apomatic genes into hybrid varieties. तो आपका ये चैप्टर कंप्लीट हो गया है. अगर आप हमारे चैनल पर नए हैं तो सब्सक्राइब करना ना भूलना, और पसंद आया तो लाइक करना. मिलते हैं नेक्स्ट सेक्शन में. बाय बाय.